Clayton, can you hear me there? Okay, good.
All right. I think we'll get started. This is actually a big moment. This is the first in-person VTLSS since everything shut down. And it's, so I'm Silke Hoff, and on behalf of the entire VTLSS committee, it's a real pleasure to introduce Carla Finkielstein. And it's really fitting that Carla is giving this talk today, because Carla was actually running the VTLSS when it shut down. So she was probably one of the last people here introducing people and so on. So a lot has happened since then. And Carla now is the director of the Molecular Diagnostics Lab of VT, in addition to running her own research. And as I think will become clear from my introduction, service to the community has always been really, really large for Carla. And if all of you maybe, I know this is a painful memory, but you know, remember the early days of the pandemic when everything looked really catastrophic. And also, it had become clear that really testing would be crucial, but the kits that came from the CDC were not really functional. And I mean, there was a huge void in what could be done and how testing could be, you know, how we could get testing quickly. And Carla really stepped in and took charge. And she was neither a virologist nor an immunologist. That was not her research area or anything, but as a molecular biologist, she certainly knew how to do a PCR. And she realized that PCRs could be important in the diagnostic. And she just set up testing for Virginia Tech, initially just in her lab in Steger, and then eventually moving the whole operation to Roanoke and fusing it with the efforts that had been going on there. But so, I mean, that was an amazing feat. And I'm not sure everyone realizes also that it's not really just setting up a test, you know, and getting the people to do that and getting to run it properly and getting it approved and everything. It's also all the logistics from getting the samples in to getting the data out. And a whole number of universities all around the countries have been trying to do that, but that was a huge bottleneck for everyone. And really, Carla managed to set it all up. So very, very deservedly, she got Virginia Tech's ProSim Award earlier this year, which is the highest award given to faculty, because that was really just absolutely fantastic work at a time when it was so needed for everyone, and I think all of us profited from that. But to give you a little bit more background on her, she got her bachelor's and PhD degree from the University of Buenos Aires, and then did postdocs working on cell cycle regulation and X-ray crystallography at the University of Colorado. First in the HHMI-funded lab of Jim Mahler, and then with Jiaqing Chen, and she joined Virginia Tech in 2005. Her own research, which she will talk about today, has a really unique angle in looking at the connections between the circadian cycle and the cell cycle, and how that has influences in cancer. And even prior to the ProSim Award, Carla has done, I don't know, absolutely incredible service. She has received multiple of the Outreach Excellence Awards of Virginia Tech. I definitely cannot cover everything. She's heavily involved in the Society for Research in Biological Rhythms. She's on the board for the Virginia Breast Cancer Foundation and the Susan G. Komen Breast Cancer Foundation. She's fundraiser for the American Heart Association and multiple other foundations. She's the Associate Division Leader of Virginia Tech's Nanoscience program. She has organized multiple international exchange programs with Virginia Tech. She's engaged in outreach to community colleges. She has recently, for example, done vaccination for undocumented immigrants. Whenever there's something that can be done that serves the community, I think Carla will do it. She gives multiple interviews to several media, or she regularly gives interviews to multiple media outlets. She gives talks all over the world. Her research has received multiple awards, including from the Virginia Academy of Sciences. And also, she has, over the years, always had very close connections with the FBRI in Roanoke, or what is now called the FBRI, but now also recently has moved her lab there to be even closer. And I should say that all of the things I mentioned, of course, come on top of research and teaching and reviewing for journals and reviewing for grant agencies and so on. And I'm pretty sure I've forgotten a whole bunch of other important things. So I'm not exaggerating when I tell you that it takes about half an hour to read Carla's CV. So I don't want to take any more time. Thank you a lot for Carla for doing this on top of everything, and we're looking forward to your seminar.
Okay, first of all, I would like to thank the committee for giving me the opportunity to actually talk to you. Can you hear me over there, guys? And I want to thank Silke for your overly generous presentation. Okay, I have a lot of people that does a lot of work for me, and it's just a team. It's not one person. So today I'm going to talk a little bit about the research that we do in my lab, which is really what excites me. And in particular, I'm going to focus on molecular clocks and how we can exploit molecular clocks for cancer treatment. So for the purpose of this talk, first I'm going to start with the typical disclosures. And we always talk about how much money we have on the stock market. In my disclosure, I'm going to say that in this talk, I will not give any credit to a pan with 5 genes that has been out there and has been very famous for the last year and a half. Okay, it has nothing to do with stock. I'm not rich, unfortunately. I will not talk either about a disease that starts with C and finishes on 19, because I'm really sick and tired of this disease. And for 1 second, I'm gonna quote Silke here, and I want you to picture yourself in March last year and think, if instead of a pandemic, we had called this disease the stay-at-home challenge, maybe we don't have any pandemic by now, okay? Well, the reality is that we do have a lot of resources to actually deal with this pandemic. Some of them, unfortunately, have become very politicized. But nowadays, we have masks and vaccines, and we need to really champion the use of these resources every time that we can. So I encourage you all to do your part as scientists in trying to explain people the best that you can why using these resources is so important. So anyway, I'm going to stop preaching here, and I'm going to jump exactly on my research now. So my research explores the connection between circadian rhythms and the cell division cycle. And before I jump into the details, I would like to give you the 101 version that relates to this talk. So circadian rhythms, and I apologize for the simplicity to Shihoko, are mechanisms that keep our physiology and behavior running in a 24-hour cycle. They are formed by 3 main components. One is an input signal. The other one, and this can be light, for example. The other one is an endogenous oscillator that usually is a suprachiasmatic nucleus of the brain, and is located at the intersection between the middle of your brain and your nose, and it's the size of a rice bean and has around 20,000 neurons. And that's my full knowledge of neuroscience of the suprachiasmatic nuclei. Okay, Michelle? What is interesting is that the third component of the clock is the output pathway. And the output pathway is usually the release of a hormone or locomotor activity. So what happens with this output pathway is actually connected to what we call peripheral oscillators. These peripheral oscillators keep our body in harmony with external conditions. This is particularly important because the function of these peripheral oscillators actually regulate, for example, detoxification and drug metabolism of very many of the components that they are giving as medicine. Okay, as you can imagine, the regulation of any of these processes can lead to diseases and disorders. For example, sleep and wake disorders, jet lag, cardiovascular diseases. I mean, in most cases, we know that, for example, nowadays, heart attacks are prevalent between 6 in the morning and 9 a.m., okay? And that also coincides with a peak of hypertension. So we also know that metabolic syndromes are associated to circadian dysregulation, okay? And depression, there are very good epidemiological studies done or running in Scandinavian countries, where they really link the deregulation of the light hours of the day and the season deregulation to circadian disorders. So what is particularly interesting from the standpoint of my lab is that a number of epidemiological studies link the deregulation of circadian rhythms to cancer incidence. And in fact, the first work was published by epidemiologists in 2007, and it was taken by the International Agency for Research in Cancer. And after a thorough analysis, they actually considered that night shift work could be a probable carcinogenic. In 2019, this assessment was reviewed, and they confirmed one more time that actually night shift work is a potential carcinogenic. In fact, in places like England and many other places in Europe that are way much more advanced in regulations that has to do with the night shift work, there's a number of years that a person that works night shift can actually work in that particular shift. We know nowadays that women that work more than 5 years night shift, they have a 37% higher propensity of developing breast cancer. And the same is for prostate cancer for men. And this is not based on small epidemiological studies. This is actually based in a large epidemiological study that started in 1976 and enrolled 280,000 women. That is called the Nurse Health Study. So with all this in mind, we run a very interesting project in collaboration with one of my friends and colleague, Diego Golombek, in which we aim to test the hypothesis that continuous circadian disruption of a person or an animal will actually lead to remodeling of the tumor immune microenvironment and favor tumor growth, for example. So we did this experiment, and we started this experiment in the opposite side of the world, in Argentina. And what we did in this experiment, we injected animals with cells that they were non-metastatic melanoma cells. And these cells were injected in animals that they were previously maintained in a normal circadian condition, light dark cycle of 12 hours, or they were jet lagged. You don't need to put a mice in a plane to jet lag a mice. Actually, all what you need to do, the only reason is because you don't get miles, but all what you need to do is actually perform what is called a phase advance of 6 hours every 2 days. You change the light dark cycle, and the animal is totally chronically jet lagged. And then after a couple of weeks, I think it was 3 weeks, we injected the animals subcutaneously with these melanoma non-metastatic cells, and we followed the progression of the tumor over a couple of weeks. What we found was actually quite interesting. First, we found that the latency of the tumor, which is measured by the time in which you inject the cells and the first time that you actually have a palpable tumor, was much shorter in animals exposed to chronic jet lag. But also we found that the size of the tumor was way much bigger as well. At this point, we needed to sacrifice the animals. You can see that the survival rate, the survival curve goes really down for animals that they were exposed to chronic jet lag. So Diego's lab is focused a lot on the immunology of circadian rhythms, and in collaboration with one of his postdocs, she actually measured some immunological parameters associated with the tumor microenvironment. She found out, for example, that the tumor microenvironment shifted from having macrophages that they are anti-inflammatory to pro-inflammatories, creating an environment that was more likely to favor growth. She also found out that the clock cells were totally deregulated in the tumor, and she also found out that very many of the cell cycle components were deregulated in the tumor cells as well, and in particular in those exposed to chronic jet lag. The most interesting part of this work that at least interests me the most is the fact that we observe that the accumulation of pro-inflammatory cytokines prime the cells from other organs, like the liver, whose cells are normally in quiescent state, to actually be competent to mutagenic signals. So the next phase of this research, which is ongoing right now, is the study of metastasis in a model of melanoma that is metastatic in nature. So how is that these cells, the circadian cells actually, or these cells that they have the circadian components relate to the processes that we actually see ongoing in the cell? Well, circadian rhythms are driven by loops that are interconnected, positive and negative loops. And all these positive and negative loops are driven mainly by one core transcription factor that is an heterodimer. And in mammals, it's called the clock-mimal heterodimer. Clock-mimal drives the expression of multiple genes, including those that negatively regulate the same complex, but also is involved in the expression of clock control genes. These clock control genes, these clock control genes are genes that, interestingly, control the expression of numerous pathways in the cell, including, oops, sabotage, these are my, so including pathways involving, for example, the DNA damage response, cell division, oxidative stress, hormone secretion, and all these pathways really control the physiology of the body. So as you can imagine, the role of the clock is tremendous. It has to not only control the timely expression of, or control the timely regulation of all these processes, but also the deregulation of any of these processes has tremendous physiological consequences. So in our lab, we are interested in one main question, and it's how cells measure time. And as you can imagine, measure of time is not trivial, it's important, especially if you consider that mammalian cells are usually resting in G1 phase of the cell cycle during the night, and they're very happy there, okay? And around 8 in the morning, 9 in the morning, these mammalian cells, they actually enter the DNA duplication phase, or S phase, and I forgot to duplicate the legs, but the legs duplicate in cells too, okay? And these guys, they go through the whole day until around 5 p.m., and around 5 p.m. the cells enter in G2 phase of the cell cycle, where check that everything is going okay, and around 10 p.m. or 11, they undergo mitosis in a process that is roughly 1 hour, and after that, it repeats the process again. So we are very, very interested in the lab in understanding how this process is so timely regulated. So how the cells sense time cues, how they adjust, reset, and reprogram their metabolic and genetic makeup to environmental alterations. How is that these cells couple the response to genotoxic stress and sustains the whole tissue collective rhythms, and a number of other questions that they are a little bit beyond just the one cell. Initially, the concept is that how is that this cell cycle and this circadian cycle are connected. And the reason because we know that they are connected is not only because there's accumulating number of molecules that they seem to be interplaying between these two cycles nowadays, is because it kind of makes sense. I mean, if you consider that these two cycles need to oscillate in harmony, they need to be connected. So which ones are the big questions that we try to answer in our lab? Well, we want to know how chronic environmental perturbation contribute to the etiology of tumors and the phenotypic plasticity of the tumors. If you think that every single cell in a tissue has its own clock, we know that the tissue works in total harmony. Every single cell needs to connect and be in harmony with the next one. How is that these clocks that they are individual in each cell connect and synchronize one to each other to make sure that all the functions, they work exactly the same at the same time? We are interested in checking whether there are different time systems in malignant cells versus normal cells, and whether we can exploit those differences in a therapeutic way. And lastly, we would like to focus, and this is where my lab is going, and that's why being at the FBRI might be a benefit for me, is because I'm very interested in time-of-day medicine. I'm very interested in moving our lab towards a lab that studies or translates basic research and provides evidences for why or which one is the best time to apply medicine for the better treatment of diseases, in particular in cancer. So if you really go back and study what's the relationship between cancer and circadian rhythm, I think the first paper that you need to refer to is one that was published in 2002 when I was still postdoc. And it really caught my attention. And the reason because it caught my attention, because it was one of those papers where the first figure was awesome, and the rest of the paper was kind of shooting in the dark, trying to find out an explanation for such an awesome phenotype. Basically, what they did in this paper is they knock out a gene called period-2, and as you can imagine, if period-2 is involved in sustaining circadian rhythm, you might expect that an animal that is knocked out in period-2 will go to sleep early or wake up late or something like that. Well, these guys, they did find that, but on top of that, they found out that the animal developed spontaneous lymphomas. And that was totally out of the blue, and I think that is what made this paper particularly hot in the field. And I think it was a fantastic observation, except that unfortunately they were pure circadian biologists and they couldn't make a good connection in trying to explain why that happened. Nevertheless, ended up being really the inception of the field of circadian cell connection or circadian cancer connection that we have these days. Later on, there were other papers that shows that somehow their regulation, or in many ways, either by overexpression or knockout or several components of the circadian pathway, they actually somehow impact different aspects of the cell regulation. So pretty much it was then when I decided that I will study how this cancer circadian connection starts, and the main question that we had really was how to identify mechanisms that connect, clock genes to proliferation, and whether the regulation of one of them is associated to one particular cancer phenotype. Now, any of us that work in signal transduction knows that working in signal transduction in mammalian cells is particularly daunting. And I think the first person that actually captured how complicated it is to work in signal transduction in mammalian cells is Jackson Pollock. Actually, you look at any canvas from Jackson Pollock, and I pick number 6 of his masterpiece is my favorite. And you can actually see that here and there are molecules, right? I mean, this is the way that signal transduction works for me. It's a connection of multiple and puzzling, pieces that eventually they will make sense, but for which we really need to work hard. That's why we need model organisms that are so simple that can help us to dissect the intricate small parts before we can put all the pieces together. Anyway, with this on mind, we started working on these connections, and the first thing that we actually did in my lab was to investigate whether what's the role of the circadian protein period-2 in controlling cell progression, if any, because at that time there was no much information except the fact that a knockout animal in PER2 developed tumors. So I gave that task to one of my initial graduate students, and he ran a number of experiments that I'm not going to describe now because everything is already published. But basically, what he did is he tried to find partners for PER2 that would make sense from the standpoint of a connection between circadian rhythms and cell proliferation or cell cycle division. On a Friday afternoon, he came to my lab, to my office, and told me, Carla, I did this 2-hybrid system, million plates, million of clones, and finally I found out that the protein P53 interacts with PER2. And you're a PI, what do? You send the kid back to the bench, right? And say, no, repeat the experiment and don't find P53, because everybody works in P53. I don't want to find P53. P53 has its own meetings. I don't want to have P53, okay? So he actually repeated the experiment, not only came back a couple of weeks later with the same result, but he actually proved me totally wrong. He found out that PER2 and P53 interact endogenously, that the interaction actually favors the stability of P53, and that when PER2 is not there, P53 is very unstable. He actually mapped the P53 binding site to be on the C-terminus domain. He mapped the PER2 binding site in P53 to be on the C-terminus domain, which is usually the domain that is ubiquitinated, but P53 is negative regulator, the oncogenic protein MDM2. So Carlo did all that work, and he published a fantastic article, and this work was in collaboration with Tetsuya Gota as well. So we were very happy with this finding. But then the main question was that, okay, so PAR2 interacts with P53 and controls P53 stability, but what else? What's going on? What do we need to control P53 stability? And then it came a couple of experiments that were also published, and I'm not going to go into a lot of details, but basically my group came up with a fantastic idea of using chimeras to actually demonstrate that when P53 is covalently bound to PAR2 or is bound in a way that it cannot be dissociated, the whole DNA damage checkpoint response is abrogated. And that was very interesting. We also showed, not only that, we also showed that later on, if you try to reconstitute the whole checkpoint signaling, and instead of using a P53, you use a chimera form that maintains PAR2 bound to P53, the downstream regulators of P53, even in response to DNA damage, are not expressed. So this suggests that when PAR2 is associated to P53, it not only controls P53 stability, but also controls P53 transcriptional activity. And this was like a big deal in the field, because it was the first time that a key checkpoint molecule was associated to a circadian factor. So we moved from not only understanding that PAR2 was controlling the stability of P53, but in conditions of genotoxic stress, and we tested different genotoxic stress conditions, P53 and PAR2 controls the transcriptional activity of P53 as well. So everybody was happy. And then we have this experiment. You know, you always have that one experiment, right, that it ruins your weekend, because it's an experiment that was perfectly well designed, reproducible, and nevertheless we couldn't explain it in the context of the model that we were proposing. And the experiment is very simple. If you claim that PAR2 stabilizes P53, you would expect that every time that you have PAR2, you have P53, right? Because one stabilizes each other. So I asked Tatsuya to run a quick time course experiment to prove that it was the case, to add it as a supplementary material in one of the papers. And then he found out, and he was always very perfect in terms of his techniques for when doing experiments. He actually found out that, you see, when PAR2 is at the minimum, P53 was at the maximum. And I was like, it's another case like, no, I don't want to see this result. What are you talking about? So this is one situation in science in which you have two routes. You either need to retract all your papers, or there is something in your hypothesis that is missing, and you need to find it, okay? So I struggled with this experiment for a really, really long time, until I have the really pleasure of presenting some of those results in a Gordon conference, and talking to one of my colleagues, Jay Kim, and came out with some kind of initial explanation. We decided that instead of running whole extracts of cells to check whether the proteins were present or not at the same time, maybe we should pay attention to compartmentalization and separate nucleus and cytoplasmic fractions and find out if the proteins coexist in one particular fraction and not the other one. And what we found is that in the cytoplasmic fractions, P53 and PAR2 were antiphase, whereas it wasn't the case in the nucleus. So basically what we were having is a situation in which we have PAR2, and then in the cytoplasms both are antiphase, but in the nucleus we're both together. So I look at him like, I don't know. I mean, I don't know. I don't even know how to start thinking about how to solve this problem. But he is a systems biologist, and systems biologists, they get all excited about these problems that we don't have any clue how to explain. And he told me, Carla, let me take into this challenge. And what we did is something that this is my way of how systems biology works. So you give a bunch of results to a systems biologist, okay, bad ones and good ones. They put everything on the computer. They come up with a model. They ask you to test several hypotheses. You fail miserably trying to do that. They don't care. You just give them the result back, and they will put it on the computer, and they keep doing this iteration until finally they come out with a model. And in fact, one day, Jay sent me an email and said, Carla, I found a solution for your problem. And I was like, yeah, we found the solution for the problem. It's in the attachment of my email. And this one was his attachment. This is me running around in the lab saying, Jay found the solution to our problem, okay? And I was like, wow, this is an interesting solution for the problem. Maybe I should try to understand it. And then this is me a couple of hours later trying to make sense of all this and thinking, man, I passed calculus with an A through the whole university, okay? And I'm not able to understand any single thing that he's talking about here. So then I called Jay, and he's in South Korea, I'm here, so we have these kind of very interesting conversations over time. And he explained me this something like 20 times, to the point that I'm not sure that I understand them. I went through a range of frustration in the middle, but eventually, after so many times, I arrived to the conclusion that I might not understand anything, but I can explain it to you. Okay? So basically, the idea is that what he did in this analysis was something very, very simple. All what he did was to put all possible scenarios in the computer by which PER2 and P53 might interact and give these results. So he analyzed how whether PER2 could influence translocation, or could influence the degradation, or could influence the stability. He put absolutely everything, unbiased, totally unbiased. And that's what I really like about the field. I mean, they don't really, I feel like sometimes we experimentalists, we are a little biased in the way that we think about our own mechanisms. He was totally unbiased, and he came with the conclusion that there are actually three reasons, or if three particular scenarios can be demonstrated, that would explain this situation. He told me that if we can prove that the half-life of P53 in the nucleus is longer than the one on the cytosol, and we can prove that PER2 actually shuttles P53 inside the nucleus, and that P53 can bind to PER2 even if there is some degree of ubiquitination, then this scenario is absolutely possible. So we did all those experiments, and we went back and forth, and this was a fantastic collaboration between assistance biologists and experimentalists, and in that proving that actually, in fact, PER2 and P53 they associate on the cytosol, and that PER2 helps the translocation of P53 to the nucleus. So slowly, we were kind of feeling the pieces of the puzzle that actually we're kind of making sense of what we were seeing or expecting to see on the cells. So we published that paper in the Proceedings of the National Academy of Science. The whole lab went out for celebration, and this is the politically correct picture of the celebration with coffee in a Starbucks. And during this celebration, a super talented grad student in my lab, Jingjin Liu, she had an idea that she has been trying to pass to me probably for a year. And I was very reluctant to pay attention to that idea because we were so focused on the shuttling of PER2 and P53 that I didn't want to get distracted with other things. And she thought, okay, this is the perfect moment for me to pass this new hypothesis to Carla. Jingjin is on her second postdoc. She did her first postdoc at the University of Pennsylvania, and her second postdoc at the St. Jude Hospital, where she is now. In the back is this student, another super talented student. He's an undergrad. He was an undergrad student in my lab, Philip Stauffer. And he is now a graduate student. He published a paper with us, and he's a graduate student in Vanderbilt now. Anyway, so what Jingjin was telling me was something like, Carla, this is a pile of paper that I want you to review this weekend. And what I did with that pile of paper is to go over and find out that the information in these papers was not consistent with the model. So the model of regulation of circadian rhythms involved the degradation of PER2 in a phosphorylation-dependent manner. Casein kinase 1 epsilon delta phosphorylates PER2, and that phosphorylation triggers the binding to the beta TRCP ubiquitin ligase, and then PER2 is degraded. And that has been the dogma, and it still is, okay? We didn't change the dogma, okay? We just bent the dogma. So I was looking at her, I was like, well, that's fine. And then she said, but yeah, Carla, but look at this doesn't make any sense. If beta-TRCP is involved in the degradation of PER2, when you overexpress a dominant negative form of beta-TRCP, you would expect to have more PER2 and more phosphorylated PER2, okay? And there is no any change. PER2 gets degraded. Not only that, there were a number of other papers, some from Chogun Lee labs. His papers are super elegant, where he actually shows that even if you inhibit casein kinase or casein kinase 1 epsilon or delta, or you have a dominant negative form expressed in the cell still has circadian rhythms. And that shouldn't happen if casein kinase 1 wasn't functional. And then there was another paper published kind of the same year in which they studied the kinetics of degradation of period 2. And this came from David Bircheff's lab. And the conclusion of his paper is that the three-stage degradation process of PER2 should involve the existence of an ubiquitin ligase that binds PER2 and is not dependent on phosphorylation. So Jin came to me and said, Carla, I want to look for that ubiquitin ligase. And I say, well, okay, there are only 600 on the whole mammalian genome. Be my guest. It's all yours. Okay? All what you need to do is to disprove one per day, and in 2 years, you'll be done. And she was like, ay, ay, ay. And she came to me and said, Carla, you forget something that is absolutely critical. We discovered that PER2 binds to P53, but also binds to the P53 negative regulator MDM2 and form a trimetic complex that exists in the nuclei. Now, she was right. I forgot about that piece for 1 second. And then she says, well, MDM2 is an ubiquitin ligase. It's not a typical ubiquitin ligase, but it's an ubiquitin ligase, and normally doesn't need the substrate to be phosphorylated. So with that on hand, she ran a number of experiments with the tremendous help of another fantastic other student in the lab, Charlene Su, that you probably heard his talk a couple of times before. Charlene is extremely dedicated to have the most beautiful experiments for publication. I feel bad when I look back to my own publications. They don't have this quality. He actually came out with a bunch of critical experiments that prove that PER2 and MDM2 not only interact, but also that the interaction leads to the ubiquitination of PER2. First, they showed in vitro that MDM2 and PER interact. They showed that endogenous interaction in cells. And then in a very elegant experiment, he mapped the epitope in which PER2 and MDM2 interact. And this was a very interesting experiment and critical because he showed that the interaction of PER2 in MDM happens in a domain that is not the N terminus where P53 is supposed to bind to MDM, or the C terminus where the ubiquitin ligase activity of MDM is located. So that is particularly relevant because the fact that PER2 is binding somewhere in the middle explains why the trimetic complex can exist, because MDM still can bind P53 and still can have ubiquitin ligase activity. With that piece of information in mind, they actually show in vitro that PER2 is ubiquitinated and polyubiquitinated, and it's not the case when we kill the activity of MDM2. Not only that, we found out that that ubiquitination happens in the same region where P53, the same region in PER2 where P53 binds. And we did that in very many ways. And one of them, we actually did some side-direct mutagenesis. This was a very demanding experiment in many ways, but we determined that there are certain regions on PER2 that they are more likely to get ubiquitinated. We mapped the region to the level of the amino acid, and then we needed to do molecular modeling in collaboration with Ambran to explain why certain lysins were ubiquitinated and not others, and it's due to a conformational shift that happened in PER2. For the record, PER2 is an intrinsically in-structured protein, and if you try to purify PER2, it's really a very difficult molecule to purify. It's over 1,400 amino acids, and it's really like an spaghetti. So when you try to purify it, it's chopped. Okay, so the biochemistry behind all the data that I'm showing you is it's very difficult, it's very demanding. So anyway, we actually show that the interaction between PAR2 and MDM2 has a direct impact on circadian rhythms. And you could see it in this particular graph right here, in which we monitor oscillations of the PAR gene. And you can see that when we overexpress MDM2, the period is shorter because PAR2 is degraded. We also knock down MDM2, and we found out that if we stabilize this PAR2, then the period length is higher, is larger. And if we inhibit the MDM2 ubiquitin activity using semperidine, which is a specific inhibitor of MDM2, then PAR2 is more stable, and therefore the period is longer. So we came out with this model that is kind of the model that we are now exploring in the lab. And as you can imagine, this model is only 3 or 4 molecules. That's why Pollock was right. There's a lot that we are still missing here. So let me move towards what I call the next phase of the research in my lab. So this is a lot of basic science. We keep doing things like this. We keep discovering pieces of this big puzzle. But we are moving to kind of in a new direction. And that comes a lot with our understanding of the relationship between these molecules and how the relationship between this molecule, what is not the way that it should be, could be somehow related to disease development. So one thing that I'm pretty sure you all know is that P53 is a tremendous tumor suppressor and is mutated in 80% of all type of cancers. Okay, it accumulates mutations crazy. And there is one particular region in P53 where all these mutations are accumulated, and that region is where PAR2 binds. Okay? Not only that, MDM2 is also a molecule that is highly overexpressed in tumors for 2 reasons. Either is located by translocation to the downstream of a different promoter, or the gene is duplicated. So another one that is highly mutation, and this is also our data, is PAR2. Well, one thing that we found is by analyzing over 3,000 breast cancer tumors is that PAR2 is highly mutated in this particular region depending on the subtype of breast cancer. And when it's mutated in this particular region, this is the region where actually P53 binds to. So we don't really know exactly whether the subtypes, this figure where the mutations in the different subtype has a different outcome because on what partners binds to PAR2, but those are things that we need to investigate. So why is all this important? Well, the reason because all this is important is because in the therapeutics, very many small molecules aim to actually treat patients that they have certain cancers by reversing the structure of P53. So P53, when it's mutated, changes conformation, and the idea is you give this molecule to P53, goes back to the wild type, and triggers cell death. That's the whole idea, okay? And there's very many therapeutics like that already, either in clinical trials or already published in the literature. So we thought, this is quite interesting. So that means that if we give a drug to P53, and it binds P53, P53 becomes wild type and the cell dies. Okay, what happens if that mutation in P53 that changes the structure of P53 is blocked, okay, is blocked on the active site by binding to PAR2. So this is a thesis of my student Jesse Janowski, and one thing that we've been working on is mutations on P53 that are particularly present in a number of tumors that lay on the area where PAR2 binds to. So this molecule right here is P53. I mean, it's actually a part of P53. This helix is where the DNA binding part takes place on P53. And this molecule that you see here is one of the domain of PAR2. I just want you to pay attention to this little loop on P53 that is here. That loop has a mutation in something like 43% of all cancers. And when this mutation exists, this loop changes conformation from this to 90 degrees down. When this conformation takes place, we find that PER2 is actually bound more stronger to P53. So it's interesting because one of the drugs that there is now in phase 1 clinical trial, Finkielstein, is a drug that binds here and changes the conformation of P53 to wild type. So we hypothesized that, well, if PER2 is bound even stronger to P53, then when you try to give the drug Finkielstein, then it won't compete off PER2, and then you won't be able to reverse the effect of P53. Yes? Oh, sorry, there was a question. So then we found we actually found that even if you add 1 millimolar concentration of Finkielstein, you cannot disrupt P53-PER2 interaction. What does it mean? It means that if you're going to give this drug to a person that has a tumor, you either need to give it to a person whose tumor doesn't express PER2 at all, okay, so then it will work, or you give it to a person in which the tumor expresses PER2 at minimum, okay, so then it can be effective. So this is kind of the idea of combining some of our basic research with some of the translational work that is ongoing now. So I'm going to spend the last 2 minutes of my talk giving you an idea of where we're going. So I think this is what I'm struggling the most these days. Can basic scientists actually help to implement time-of-day medicine? Okay, and why is this? I think we have a lot of information out there that there is a clear connection between many circadian components and components that control the outcome of processes involved in cancer. So can we pick the best time to actually give those drugs to patients so that we can have more effective therapeutics? We are in times in which we focus on personalized medicine, and this is particularly true for cancer. Okay, nowadays, a person that has cancer has their tumor removed, and on top of the biopsy, you can have your whole genome analyzed. Okay, and based on that is the therapeutic that is given to the patient. So we went from one-size-fits-all to some kind of personalized therapeutics that has been really way much more successful. Okay, but one thing that is important, and I want to bring to your attention the work of one of my colleagues, John Hoganesh, who has been very successful in implementing chronotherapeutics in the clinic in the Cincinnati Children's Hospital. If you look at each spot here is a clinical trial that was done since 1965 to 2020. Okay, and these are clinical trials in which a medication was given at 2 different times of the day, same medication at 2 different times. And you can see that in very many of these diseases, particularly in cancer, giving the medication at one particular time of the day had better outcome. And this is particularly true for medications like chemotherapeutic drugs, that they have different half-life, okay? And you don't want to have a patient experiencing an overdose of chemotherapeutic drugs because of the side effects of the chemotherapeutic drugs. In the cancer field, many patients die of cancer, but very many other ones, they quit the therapeutic because of the aggressiveness of the therapeutics. So the question is, can we actually deliver drugs, chemotherapeutic drugs, to patients at time of the days in which the drug is more efficient, meaning that the target is there? So then the treatment is more efficient for the patient, and the quality of life of the patient is much better. So this is what we really aim in our lab to do. And I'm going to give you an example of where we are in the world. There are roughly 220,000 clinical trials ongoing in the world. You can go to the clinicaltrial.gov website and check that. Okay? Of these 220,000 clinical trials, only 348, okay, involves any kind of therapeutics that involve a circadian intervention. So only 348 out of the 220,000 are checking whether the time of day medication is influencing in any way on the outcome of the disease. Not only that, we actually know that of that 0.16%, only 3% of those therapeutics are treatments that are associated with cancer. However, very many of the chemotherapeutic drugs that we use these days, they have targets that in one way or another one are circadian regulated. And on top of that, among the 100 best-selling drugs in the market, and you can pick the one that you like the most. Anything from Ritalin to Viagra, they all target circadian molecules, which means that if you take it at the wrong time of the day, you might not have the maximum effect, okay? So this is just an example, and this is from John's paper, where you can actually see which ones are the target genes for these very many different molecules, and based on their half-life, which one will be the best one, the best time to deliver them. So I'm going to leave you with something that I read very recently that was discouraging, but I hope we can change. Only 4 of the 50 currently most prescribed drugs that have FDA-labeled time-of-day dosing recommendations. Okay, and these drugs are actually for hypertension or asthma. Okay, nothing else. So where the lab is going, we want to emphasize point of control in circadian biology that intersect critical nodes that can be used for can be exploited for therapeutic purposes, and I hope we can provide molecular basis to our colleagues to consider time-of-day medication in their clinical trials. I'm going to finish by thanking everybody doing the job, and this is a snapshot of all the people that has been over these years in my lab, and I feel sorry if I didn't put you there. It's a little bit unfair. But I didn't have a space. I have more than 120 students, undergrad students rotating or doing research in my lab. Very many of them publish papers, and they've been accepted in universities for graduate work or in the health professions in many ways. I've been very lucky to have fantastic graduate students that have been passionate and at the same time super intelligent, much better than me. Then came out with crazy ideas that turned out to be really interesting stories. I have a new cohort of students in the lab now, and I welcome all the new graduate students, Siki, Lisa, and Jonathan. And Jesse is not considered new anymore because he has been in the lab for almost 2 years. And all the new undergrads that are doing a lot of really great job. I want to thank my collaborators, Jay Kim, John Tyson, and Brown, and Diego Golombek, who has been instrumental in all the work that we did in my lab. And my other lab, this is the crew that runs the molecular diagnostics lab, a fantastic group of people that is incredible the way that they work and the commitment that they have to make sure that we can all do our job every day. With this, I'm going to finish, and I'll be happy to answer any questions. Okay, this is not good. Usually when the people don't ask questions, it's because my talk was very confusing. Or what? Yeah, go ahead.
Considering that some drugs are most effective at like inconvenient times, what are some examples of like delayed delivery methods that wouldn't patients circadian?
Yeah. Your question is really good. What's your name?
Samir.
Samir. Oh, Samir.
Yeah.
Good. Finally, nice meeting you. So this is the thing. Ideally, you want to have the drug delivered at a time that you know is going to have their peak on the bloodstream when it has to have the maximum effect, right? Now, what happens if you need to take that drug 3 in the morning, for example? That would be a problem, and that's usually the problem that, when I talk to clinicians, that's what they tell me. It's like, Carla, how do we solve this problem? And I say, well, that's why we have nanotherapeutics. So maybe what we need to do is to give the drug to the person, maybe at 8 p.m., but have a delivery system that has a slow release. So the accumulation of the drug happens at 3 in the morning, but you don't need to take it at 3 in the morning. Now, there's another problem. The problem is for radiation therapy, to be honest with you, because radiation therapy is more efficient if you deliver it at night. Okay? There's a lot of research already ongoing that shows that cells that they are irradiated, synchronized and irradiated at night, they die much better than if you irradiate during the day. Now, all the radiation facilities in the hospitals, they run during the day. Okay, they don't really care that your cells are on S phase or G phase or whatever, right? You just get an appointment whenever it's open. So how are you going to change the system to actually deliver radiation at night. That is really complicated. And on top of that, you need to consider that, you know, maybe the person that runs the unit is not at the peak of their fitness at 3 in the morning to actually deliver radiation to a patient. So I don't think it's going to be something that is going to be super easy to implement, or we will need to find ways to implement. But for example, what John Hogeness did in the Children's Hospital is something that is absolutely fantastic. You're going to see it published very soon. He has a really fantastic charisma, and he managed to convince the board of the hospital to actually do a very simple change, to change the light intensity in the ICU unit and in the neonatal unit. And he proved, since the moment that that light change intensity was changed, the beds are empty much faster. Okay, so basically you're saving $20,000 per bed per day. Okay, so so that is a very easy way to implement, you know, therapeutics in a way that can help. You know, the other thing is that you cannot implement chronotherapeutics for everything, right? I mean, if your drug has a 16 hours or 17 hours half-life, well, forget about chronotherapeutics. There is no point, right? But if your drug has few hours or few minutes, then it's important. Okay, and there are some success stories. Drugs for hypertension are taken at night, and one of the reasons is because the peak of hypertension is 7 in the morning, and the heart attacks are from 6 to 9 a.m. So if you can reduce the peak of hypertension, then the number of people that are exposed, or the number of people that experience heart attack are much lower. Okay, now, you know, it's still a lot of work that we need to do, but I personally think for cancer patients it's a big game changer. Okay, yes.
I have 2 questions, Carla.
Go, Julia.
So you looked at these interactions between P53 and HER2 and then mutations in HER2. Did you look at mutations in P53, how those might affect these interactions?
Yeah, we are.
Which are predominant in cancer.
Yeah. We actually did both things, and unfortunately I couldn't show this here, but we do have a lot of protein-protein interaction studies done in which we mutate P53 and we mutate PER2, and in some cases we mutate one or the other one. And yes, we see all kinds of different things. We have, for example, situations in which just simply the mutation in P53 completely abrogates the binding of PER2. That is rare because, like I said, PER2 is an intrinsically disordered protein and really wraps around P53 and takes different shape. But there are some mutations in P53 that completely abrogate that binding. Now, what's the consequence of that? I don't know. I mean, Jesse's thesis is all about looking downstream of that interaction and see whether signaling pathways are segregated depending on what those mutations interactions take place or not and in what cell type. But yes, we've done that. The other thing that you need to consider is that those mutations usually affect the structure of the protein, right? There's a lot of structural changes associated with those mutations that affect interaction. Yeah.
Okay, and the other question is, so I'm wondering if there are studies that show, I mean, you're talking about the impact of light and intensity. How about, and just mentioned light intensity, how about frequency? And I don't know if you're aware, there are studies, this is pointed out frequently, blue light versus yellow light. Blue light increase awareness, so it's good to have it during the day, it's not good to have it during the evening.
Exactly.
So is there anything research going on there?
My understanding, and Shihoko, you can add on this, is that I'm not an expert on light, but my understanding from what I read on the literature is that controlling the ratio of blue light and red light has been critical for. That's why the intensity of the light on the hospital is all monitored based on the quantity of blue light and red light that is given to the patient, and that's why all the devices that you have these days all aim to have more red light at night, so you don't block the release of melatonin and you can still go to sleep, right? I mean, that's the big problem here. The problem is that if you're exposed to blue light, you block the release of melatonin, and then the person cannot fall asleep, right? So there's a lot in our society on educating on how to use light in a way that is mindful, so doesn't disturb our circadian rhythms. Yes, Chip.
Well, I want to go back to one of the early images that you showed in your lecture, which was where you had the mice and they had a disrupted circadian rhythm prior to injection as well as after injection. And approaching that sort of simplistically, it seems like it was also possible to design an experiment where you would have only half of that the light treatments be disrupted, so you could have a uniform light inject the cells, disrupt their cycle afterward, as well as vice versa, disrupt their cycles prior to injection and then have normal light afterwards, in a sense to perhaps separate out certain components of the process of tumorigenesis?
Yeah, yeah. The reason because the experiment was designed that way is because you know, you cannot answer all possible questions, right? We just try to answer one simple question there, and it's whether the immune system is totally disorganized in response to this chronic jet lag. We have a number of other experiments ongoing in which we don't use actually those particular mice strain. We use some in which they are genetically modified, in which we can induce, for example, the cancer to turn on, the cells to turn on and become cancerogenic at one particular time. Okay, but in that experiment, we didn't really want to put too many variations, because if you look at the original paper, which is a monster paper really, just with that simple change of 6 hours advance of light, we found so many parameters that they were totally deregulated, that if we added another component like, you know, this regulation for X amount of time, then injection, then dysregulation, I mean, it would be too much for the initial question. I mean, that's what I'm trying to say. I mean, I think what we tried to answer was something very simple, in the sense of, is something going really wrong that allows the tumor to progress faster? But we have tons of other things going on right now on that project. So that's the reality. It's just, plus the number of animals grew exponentially during the whole study, and my collaborator wasn't happy every time that I asked a question. More animals. Okay. Anything?
A lot of people have more questions on their mind, but in the interest of time, because there's so much going on today, I think people need to move on. I think this was a great proof to a very festive weekend at Virginia Tech, so thanks again, Carla.
No problem.
Carla implemented that every visitor at the VTLSS gets this.
Yeah.
So Carla, yes.
I know.
You know what's in there. And students who meet with Carla now, you'll be meeting in the room just opposite side from here, and then you can go outside. So thank you very much.
Okay.
Thanks.
Okay. Was understandable? Okay. Thank you. Totally. Yeah, I know. That's the part that I want to show today. You notice that I didn't put any information on mutations or anything. Yeah, it's quite interesting because depends on the subtype of cancer, the mutations accumulate in a different region. And for example, one has to do with Wnt signaling, for example. I didn't mention that.
Yeah, I was wondering whether you can rationalize this together with, you know, the phenotype you have in the tumor.
Yeah, exactly. And you can rationalize with the embryological pattern. Okay, so like the ones that they derive from epithelial, or they have a different set of mutation in different part. Some of them I don't know what they mean. Another thing that is quite interesting that I didn't show is that when you map mutations based on geographic regions, The mutations in PER2 in, I don't know, Asia, Latin America, and Europe, and Africa are in completely different regions. It's super interesting. I don't know exactly why. You know, I want to give that information to someone that knows more about circadian rhythms than me. But it's.
I mean, but this is really nice. I mean, there's so much there, right? I mean, this is a perfect way of making profiting from all the data that's there.
I know. In the mutation part is really cool because we see, for example, some mutations completely change the conformation of the protein. Some other mutations, they don't change the conformation. They don't seem to affect the transcriptional activity, but the protein localizes in a different place. It gets always trapped in one place. It's not even the cytosol, it's in one organelle in the cytosol. So, I don't know, there's a lot to do out there. I mean,
It's so complicated because you're really thinking about both sides. Right? There's the carcinogenesis side and there's the treatment